Apeksha Awale is a fifth-year Ph.D. candidate in the Electrical Engineering Department at the Cullen College of Engineering. Her major advisor is Dr. Jack Wolf. She was born in Mumbai, India, the city of dreams. Brought up in a free-spirited environment, her role models are her parents. She graduated with a bachelor's degree in electrical engineering in India and moved to Houston in 2010 to pursue a Ph.D. and her passion in engineering. She's expected to graduate in December 2015. She believes in the saying, if it doesn't challenge you, it doesn't change you. Allow me to present Apeksha Awale, who will introduce us to a novel tool for probing the brain to understand and precisely manipulate activity within brain circuits.
Thank you, Dr. Patrick. I visited Vegas a couple of weeks ago. People were dancing, enjoying the music, with beats that were pounding and drinks that were delicious. But this is okay if it happens once in a while. But if it keeps repeating and if it becomes a habit, we all know that there is a problem. But what if I told you that there was a way of turning this urge of drinking off just when you need it? Or maybe activate the urge of eating only when you're close to healthy foods. That way, you will never ever have to worry about overindulging on junk ever again. As an illustration of this concept, watch how this mouse responds to the presence of blue light. When the light is on, it has this immense urge of overindulging in the cheese in front of it. But when the light goes off, it has lost all that interest and it has gone back to being normal, exploring its surroundings, just until the light is on again and eating behavior resumes. Wouldn't you call this as a light-gated switch? Now, some people might argue that's easy because neurons communicate with each other with electricity. Why not just shock the brain? That sounds easy, but it is not as safe and not even as selective. Let me explain. If I'm using electrical stimulation in order to, say, activate an area of the brain which is responsible for eating, I might end up shocking a lot more than that. Some other neurons around it that are responsible for, say, movement. Look at this oversimplified picture of the brain. 3 different neuron families in a different color each, intertwined with each other. For our example, let us consider the blue ones are responsible for walking, the green ones for sleep, and the red ones for hunger. Now imagine an electrical probe with electrical stimulation, with absolutely no selectivity, trying to probe this area of the brain. It is going to activate random sets of neurons, and we all know that this is not going to go well. So there has to be a better way, and fortunately, there is. In 2005 at Stanford University, 2 scientists, Carl Dizorath and Ed Boyden, first demonstrated optical stimulation, as opposed to this electrical stimulation, which has conventionally, yes, been used. You can call it as a way of manipulating only specific nerve cells just by using the power of light. Now, neurons are usually not sensitive to light, but you can make them sensitive to light by using a new technique they discovered, which is called optogenetics. The way this works is that you take a light-sensitive protein from some sort of an algae, construct it with a carrier, inject it into the brain. Let the neuron synthesize this new guest and let it sit in there for a while. Now, if you go with light into the brain, only those neurons which have synthesized that protein will respond and get activated. So let me go back to the previous experiment that I just did, but this time I'm going to use optical stimulation. Now, with optical stimulation, I can only activate the neurons which triggered eating behavior, while leaving all the neurons around them absolutely untouched. So in the case of that mouse that you saw, it was infused with proteins that were responsive to blue light. And then that blue light was used in order to manipulate and regulate that eating behavior. Now, all of this is fascinating. It's science. It is a great technique. But we need the right set of tools in order to implement such an idea. And here in the electrical engineering department, we are making one such tool. It is called an optrode. So let's see what we need. We need 2 things, light delivery and reading of neuronal signals, which are essentially electrical impulses. When you think of light delivery, wouldn't you think of an optical fiber? And that's exactly what we are using for the light delivery part. And now I know what all of you are thinking, okay, this has to be implanted in the brain, so it has to be incredibly thin. So let me compare the size. The average size of a human hair is only 80 microns. And the probe that we're using is only 50 microns. So that is incredibly thin to work with. Now the probe does the light delivery part. But what about reading of the electrical signals from the brain that the neurons are trying to communicate with each other and with us? For that purpose, what I do is I take a spray paint of a metallic element, like gold in my case, a stencil, and I paint these conductor lines along the length of the probe. And this is kind of how it looks like once it is all ready and done and finished. In order to manufacture these amazing devices, we have a fully functional manufacturing center here at the University of Houston. Each of this equipment is responsible for a single process, and we have developed and designed a constructed process flow that we follow in order to manufacture our devices. So the light comes out of the tip of the optical fiber. It activates the genetically infused neurons around it. The recording sites read those electrical signals, and those signals are transferred from the tip to the very back end using the conductor lines. Now observe how there are panels on all the four sides, and that's so that you could read from a three-dimensional space, because we all know that the brain is the most weirdly wired thing in the entire universe. Now when you're looking to deliver such a reliable, fully functional device, a lot of factors come into play other than just reading neuron signals. So our probe is very inexpensive. It can read in a 3D space like I just showed you. It is flexible, and you can scale the size of the optical fiber. It is long enough for deep brain stimulations, for reaching deep brain structures, and at the same time, it is thin such that it does not cause any nerve tissue damage. And finally, you can use it for a matter of days or weeks without any observable degradation. The recording setup consists of the probe interfaced with the laser source for the light delivery part. Once the light has been delivered, the neural response is read as a function of spikes per second. Every blue flash that you can see here is the on time of the laser. And watch how the neural activity rises at a very high rate with every flash of light. With all this said and done, let me go back to the drinking problem or that healthy eating habit that we were talking about. We could tackle those kind of problems by using these ideas, this tool, this technique. As a matter of fact, Optogenetics has recently been approved by the FDA for testing in human patients. And if you ask me, this is a great advancement. Because let's look at it from a broader perspective. You could activate photoreceptors in a person suffering from Parkinson's disease, suffering from a blindness disorder. You could activate hand or leg movements in a person suffering from Parkinson's disease. On the same lines, in 2013, President Obama introduced the BRAIN Initiative. His goal was to study what he calls the 3 pounds of matter that sits between our ears. We have been very fortunate to receive a grant under this initiative to manufacture our Optrode. So let's take a moment. With 100 billions of neurons and 100 trillions of their connections, the human brain has been one of the greatest mysteries in the field of science and medicine today. To unlock that mystery is a challenge that we have accepted. What we need today is a dynamic picture of the brain in action so that we better understand how we think, how we learn, and how we remember. The need for new treatment of neurological diseases right now is so high that there could not have been a better time for undertaking this challenge. Thank you.